Hello everyone, I'm Zijian Feng from the MPI CBS Leipzig, and my topic is about evaluation the TMS-induced heart-brain coupling. So as we talk about a lot in recent days, the anatomical framework of the heart-brain connection is like that, mainly about the anatomic nerve system. And the sympathetic nerve system is about increasing the heart rate, and otherwise the parasympathetic way is decrease the heart rate. And the vagus nerve in the parasympathetic system is a very crucial component to decreasing the heart rate. Well, the empirical findings show that the stimulation of the vagus nerve results in a very immediate response, just typically within the same cardiac cycle, and it's very short-lived. It's about 1 to 2 heartbeats, and after this activity decrease, the heart rate returns to normal rapidly. So this allowing for quick verification of the optimal target finding. And in patients cohort, there's a bunch of reported that the elevated heart rate and reduced heart rate variability are very common in the depression cohorts. From the theory of the ESAGers, we can see that the frontal vagus nerve pathway, and there's overlapping hubs in depression network and the brain axis, including the key nodes of the DLPFC and SGGC, as well as the vagus nerve. So many people following this theory did some RTMS study, and they've reported that there's a heart rate deceleration, and this would mediate it via the frontal vagus nerve pathway. However, there are some challenges. For example, you can see here there are there's a set specific heart deceleration following the RTMS stimulation. For example, the FC4 and F4 areas has some effect on the heart rate deceleration, but the surrounding areas like C4 and these areas has the opposite effect of the RTMS. And furthermore, there's no consistent conclusion on the clinical symptom associated with the heart rate reduction. And recently, there's an advanced protocol named NCG 2.0. It is a 10 Hz dash protocol, and there's a new measurement named heart-brain coupling. So what's that? We can see here the vertical line is the RTMS twins, and then after this twins, there's a heart rate deceleration after each block. So the entrainment of this stimulated frequency represents this green line is the heart-brain coupling. So it's the power of the stimulated frequency. And the further step, they did FC-guided RTMS with 5 types of the FC guiding methods, and they reported that there's a significant effect of the negative SGCC guided compared to the neutral SGCC functional connectivity guided RTMS. However, we can see the detail. There's a significant effect of the left frontal area and as well as the left parietal stimulation. But the left, both the left parietal and the frontal areas connectivity has no significant correlation with the heart-brain coupling power. However, there's a significant positive correlation between the another hemisphere with the both the right frontal as well as the right parietal negative connectivity with the SGACC. So the question is that, is this robust, and does it really work with the heart-brain coupling with the NCG TMS 2.0? So we did a replication study first. There are 20 participants in our cohort, and we had 7 targets totally. The beam F3 and 5 centimeters. These two are the traditionally anatomical method guided of the depression RTMS treatment. And 4 other surroundings target, which means that 2 centimeters anterior, posterior, lateral, and medial of the F3 target. And this is sham-controlled stimulation. So for the replication, we did 3 times of the RTMS protocol. And I'll introduce the details of the 10 Hertz RTMS protocol. We can see here, this is a 10 Hertz protocol with 5 seconds stimulation on, 11 seconds stimulation off, which means that the cycle is 0.0625 Hertz of this protocol. So the hypothesis is that when the stimulation on, the heart rate decreased, and when the stimulation off, the heart rate back to normal. And furthermore, this is a sweep intensity study. So the TMS has no consistent output during the whole protocol. At the very beginning, for example, this is the first block, is no intensity as the baseline. And then it's a sweep twins. This 15 stimulation twins with 2% machine output increments up to the maximum of the 120% motor threshold. So this is the result of what they did. They just showed individualized result. You can see here, when the higher intensity, there's a good heart-brain coupling of this specific participant. Well, they showed all the 10 subjects of this cohort trajectories. I would say it's messy and chaotic. Once you do some group analysis, I don't think there's a very consistent result of this figure showing. And this is our results of the 2 sessions in this whole group. We can see that actually the sham and 5 centimeters line is similar to this messy plotting once we zoom in a bit of these lines. And what we found that the F3 and F3 anterior, as well as the lateral and F3 posterior, has some effect during the higher intensities, as we can see in this plotting. And this is an example of the within subjects plotting. We can see there are 3 sessions with a whole 7 targets. And the right corner showed the Spearman correlation of that. I would say most of them has lower than 0.3 correlation coefficient, which means that the replication or the reliability across these 3 sessions is not really good. And this is the group results of the correlation. And we can see that the within targets correlation is quite low, and most of them are lower than the 0.3. And this is the replication measurement of the ICC index. We can see here, by the way, this index should be like over 0.4, which means that there's a moderate replication. So according to that, we can see here, during the higher intensity, there are some moderate blocks of the certain targets, is the F3 anterior and the F3 lateral. So after that, we also investigate, is there any true effect of the TMS-induced heart-brain coupling? So we use the linear mixed model to model all the 7 targets with all the intensity. Through the results, we can see that as we hypothesized, the sham has no any effect as the baseline, and the F3 anterior and the F3 lateral has some significant effect during the higher intensity. So there are some take-home messages. We replicate the NCG 2.0 protocol in a larger healthy group. And at the higher intensities, these 4 targets showed stronger, higher heart-brain coupling. And the anterior and lateral F3 demonstrated moderate reproducibility. Well, we can see there are some like intro and individual variants of the individualized subject plotting. That's all. Thanks a lot. Oh, sorry. I have to thank my team.
Questions for Zijian? Sorry.
The stimulation you applied are quite frontal, and since I participated into experiments with frontal TMS stimulation, like the muscular twitch are quite a thing. So how can you reliably differentiate that in a sham from a sham? Because like participants can really perceive when the stimulation is frontal over threshold, right?
Yeah, thank you for the question. Actually, we are using a sham coil. The sham coil has the similar type and the sound of the active RTMS, but there's no any true E field of this local field. So this sham coil cannot induce the muscle twitching.
Yeah, so far so good, but the point is that participants that receive frontal stimulation could still have muscular twitch and understand exactly that they are not in the sham.
Yeah, I get it. Yeah, exactly. I think this is the limitation of the RTMS sham controlled. Okay. We cannot avoid this situation, actually.
Yeah, but if I can say something about that, so in these protocols, you see the heart like in real time. So of course, it's not ideal control, but you know that it's happening something because you see the heart changing in real time, the heart rate. Oh, yeah.
I would say this would much more effect on the EEG or the brain signals, but for ECG, it seems okay.
Yeah. Any other question? No. I have a very so I didn't. Your replication was healthy, right? Healthy participant. Yeah, it's written there actually. No, I was wondering if you replicated also the changes in the symptoms, but it's healthy. So yeah. Thank you very much.